angels. There's worse here than angels.
I beg to differ, sir. Let it go. Well, it can't let me go, sir. Can it not while you're looking at it?
Can't stop looking at it, it'll kill you.
No, it's too tight. You have to leave me, sir. There's nothing you can do.
You don't believe.
Yes, I'm there. Before you go,
I'm not going.
listen to me. It's important. You can't trust her.
Trust you.
What if so? You think you know her, but you don't. You don't understand who or what she is.
Tell me.
Told you more than I should. But me, you have to go. It's your duty to your friends.
Just tell me why she was installed, Cage.
She killed a man. A hero took a name. You don't want to. You don't, but you have to leave me.
Goodbye.
I would die in the knowledge that my courage to deserve it.
You should know me better.
You know me at my best.
Ready?
Content.
After being revived on McCracken, Gilliman is confronted with 10,000 years of the Imperial's decline. Determined to seek clarity and guidance, he embarks on the Terran Crusade, a perilous journey back to Terra. Upon finally reaching Terra, Gilliman is greeted not with the glory of the Emperor's presence, but with the stark reality of the throne world's dire state. Mortarion mocks his loyalty and the Emperor's perceived indifference, taunting him about their father's true nature. The two brothers face each other on lax to settle their scores. In the midst of battle, Gilliman lies dying, overcome by Mortarion's plague. The earth shakes, and reality trembles as the demonic guardian Nurgle begins to fade. Mortarion, shocked by the impossible resurrection, watches as Gilliman's armor of fate reignites, its systems blinking back to life. The demonic inhabitants flee in terror as Gilliman, glowing with a pure white light, rises from the ground. The Emperor's sword materializes in his hand, blazing with the fire of 1,000 suns. Gilliman declares his divine purpose, wielding the Emperor's power to banish Mortarion back to Nurgle's realm. Gilliman's voice echoes through the warp, declaring his loyalty to the Emperor and promising retribution.
This is one of the coolest living rooms ever. It is fully robotic. Look at that. You can move all the furniture you want. They have a nice movie theater. What does it even have? They don't have to do a single thing. But how do I get? Now, this invention is really smart. Look at this. You just scan words, and then it pops up on your computer somehow. No way you can scan. It's like writing. It actually scans. Wait, that's it. This is one of the cool. Probably the most controversial robot that we have had here at MHRL. They are able to run two different robots each way. We pull 3 pounds, but a normal 3-pound robot is allowed. How can a robot like this pull exist? Well, now you can. At MHRL, you get a weight bonus if your robot drives around the arena in a non-conventional way. In Loophole's case, it uses the shuttling system to walk around the arena. Because of that, they get 2 extra pounds. In addition to those 2 extra pounds from being a shuttler, Loophole also runs a minibot, a second robot on their squad. This minibot is done with an additional 1 pound. 3-pound robot can be 6 pounds. Meanwhile, Loophole, their main robot, 3.1 pounds. And they bring in 3 pounds like now that sounds like a recipe for a cold espresso.
పుచ్చుకుంటే ఇవి నెత్తులకి ఒరే ఎన్ని కాయిన్స్ ఎక్కడెక్కడ పడుస్తారు దా చెంపలు పగులుతాయి, ఇద్దరు చెంపలు పగులుతాయి, ముందే చెప్తున్నా.
Sadly, when you may have a genius level IQ, you have no control over your emotional state. That's why you're lashing out at here, defensive, allowing your rage at yourself to manifest in boorish, childish behavior. He has no idea why he had to resort to brute force, so he doesn't get to work here.
Take your feet off. Get the fuck out of my office. Mark, we need to talk to you about your breakfast test. You've got a perfect score. 96. Come along now. Mark, you and the other perfect scoring superstars are exempt from taking the actual test today. We are throwing you a peach party at a bowling. Well, it's down early. Come, helicopter away.
Check it out! Mark's a superstar too! Yeah, awesome! This ain't no helicopter. This is Con Air.
Take it away, Willie. Enjoy your flight. Brilliant plan to remove all the underperformers for test day, sir. Why, Seymour, and she put up my sunglasses on the phone. Well, I just retrieved them. Yeah. Oh, sir. So long, superstars.
Times actors weren't acting. This scene in Avatar, this girl gets annoyed at Jake for not being able to pronounce Nari. Nari. This smack was actually the actor's real reaction in Spider-Man Homecoming. There's a scene where Tony Stark goes to open the door, but Peter Parker
this one is shiny. It must be more valuable. And what do you think? I do know you have rarity, and I think that's something to do with these genital molecules. They could either also probably more valuable. Sapphire and value makes your stuff complex. So I'm going to say this is it's what I would have said before Morris chicken. Oh no, this was like depends on the carbon then. Now carbon has gone down almost to the back, and therefore it will still a couple of cents more. So let's try it over to the you guys have Lord of the Rings magic card? There is still anything on the franchise now they call it Universe Beyond. I wonder. Have you ever opened the magic? Bottle of water. Okay. Good evening, everyone. We'll be starting now. I hope my PowerPoint is visible, right? The PowerPoint is visible, right? Okay. Good evening, everyone, again. This is the week 9 PMRF-NPTEL session on animal physiology. And this week is titled Neural System and Special Senses Part 2. In this particular lectures, in the lectures of this particular week, we have gone through the functioning of the eye and how exactly the rod and cone cells function, as well as the anatomy of the eye in depth. And we'll be covering those in the assignment questions today. So, briefly introducing myself, my name is Anubhav Chatterjee. I'm a Prime Minister Research Fellow and PhD scholar at the Department of Biological Sciences and Bioengineering, IIT Kanpur. So, as you would know, how we do these sessions is that we will go through the assignment questions of the previous year's run of the course and try and understand how exactly to answer them and have a better understanding and grasp of the concepts in the videos themselves by doing so. So, just a reminder again that all of these sessions are recorded and the videos for these recorded sessions are uploaded to this particular playlist, and you can go ahead and watch them at your leisure if you have not been able to attend the session itself, or you have some doubt that you want to clear. And any doubts you have, you can put into the NPTEL portal itself. There's an option there, or you can put into the YouTube comment list, and we can go through those as well. So before starting, I would like to ask that if anyone has any questions on the previous or this week's course videos, if anyone has any questions, that we can go through them first and then start. Does anyone have any questions from this week's lectures? Okay. So we'll just start right off. So how we do this is that I will show you a question from the assignment and ask you and try and answer this by typing A, B, C, D, whatever you think is the correct answer into the chat, and then we will discuss the question. So the first question is, which of the following is not part of the eye?
The options are retina, cornea, iris, and glomerulus. So we have a number of answers coming in. Vishwajit and Achal both say it is glomerulus. D. Jagruti and Shantipriya also say it is D. Glomerulus. Anita also says it is D. Glomerulus. Okay. So similarly, we know a bit about the different parts of the eye. But I have a video here that will show us the anatomy of the eye in detail. So we'll go right into it. Just let me know if you can hear this.
Our eyes enable us to see all the beautiful things around us. In this video, we're going to see the structure of this eye.
Is this audible?
Yes, sir.
Okay.
The things that make up our eye. When we look at our face, it looks somewhat like this. This is my poor attempt at drawing a face. But if we were to concentrate only on the eye and get rid of all the skin, then we will be left with a ball. And that ball is usually called the eyeball. Now, let's look at this eyeball from the side and let's assume it is transparent so that we can see inside. Here it is. So we'll look at the different major parts of our eyes and we'll also see what each one does. So let's start with the one that is most visible to us, this one, this structure. This is called the iris. The iris. And when people talk about the color of the eye, they're actually talking about the color of the iris. So when light hits our eyes, the light that falls on our iris gets reflected. And the color of the light that gets reflected totally depends upon the pigment that makes it up. In this case, it is green. And so we would say that person has green eyes. In my case, for example, it is brown. So my iris reflects brown light. And so my eyes are brown in color. But notice our iris has a hole in its center. And that hole is also given a name. It's called the pupil. So let's just write that down somewhere over here. That hole is called the pupil. And as you can see, the pupil is through which the light enters into our eyes. So through the pupil, light will enter into our eyes. And this is what enables us to see things. Now, our iris has a very important job. Its job is to control the size of this pupil, the hole. And that's because during the daytime or when the ambient lighting conditions are very bright, then there are a lot of rays of light that are hitting our eyes. So we wouldn't want too many rays of light entering our eyes because in that case, that might damage our cells. So in such a case, what we'll do is our iris will make its hole smaller. The pupil becomes constricted so that it only allows the required amount of light to enter our eyes. On the other hand, if the ambient lighting conditions are very dark, so let's say it's evening time or night time or you're inside a dark room, now there won't be many rays of light hitting our eyes in the first place. Now we would want to open up that pupil, otherwise we won't be able to see anything. And so in such case, the pupil opens up, the hole opens up, allowing the light to enter. And you can do this experiment at your home. Just stand in front of a mirror in a dark room. Your pupils will now be dilated. And just flash light into your eyes and you will see immediately that pupil will get constricted, become smaller in size. Alright, now let's get rid of the iris so that we can see the other parts of the eye. So now we are only seeing a section of the iris. It's the same iris with the hole in between. Alright. So let's look at the other parts. This bulged part of the eye, which is in front, is called the cornea. So this is called the cornea. And over here we can see a lens. This is called as the lens of the eye. So it's just called the lens. Let's write that down. This is called as the lens. And the space between the cornea and the lens is filled with a watery kind of a liquid. And it's called the aqueous humor. It's called aqueous because it's watery. And if you look at this carefully, we can now see that this cornea along with the aqueous humor and the lens, they're all convex shaped. So the cornea and the lens together form a converging lens system. And so whenever we want to concentrate on a specific object, their job is to make sure that the light from that object gets focused right at the back part of our eye, because it's the back part where we have lots and lots of light sensitive cells. So the back part of the eye is completely covered with light sensitive cells, which we are seeing over here in red. And so this whole thing is also given a name that's called the retina. So let's write that down as well. So let's write that down. This light sensitive cell covering, cell lining, is called retina. And so in order to see anything clearly, the light from that object must get focused exactly on the retina. If it doesn't, it'll look blurred to us. And what do these cells do? Well, these cells, once light falls on them, they convert light into electricity. And finally, those electrical signals are carried from the retina all the way to our brain through some nerves. So there'll be some nerves that carry all these signals. The nerves will connect to all the cells of the retina, and all these electrical signals is carried to the brain. And this bundle of nerves are called optic nerves. Optic nerves. And then our brain, the command center, receives these electrical signals, does a lot of complex processing, and it's eventually able to figure out where those light rays came from, and then it constructs the image of the world around us. The whole thing is super complicated. Our brain is super complicated, but it's also pretty amazing how it's able to do all of this. And by the way, the space between the lens and the retina is filled with a jelly kind of transparent substance called the vitreous humor. The word vitreous means looks like glass or kind of like glass. Of course, it's not made of glass, it's made of organic substance. But just like glass, it is pretty transparent. So it's a jelly-like transparent material, and its job is to maintain the shape of our eyes. Without this humor, our eyes could get easily crushed under the weight of the stuff that is on top of it. So the vitreous humor is a transparent jelly-like substance. The aqueous humor is also transparent, but it's watery kind of substance. And the last thing we'll talk about are these fiber kind of things that we see over here, which are keeping our lens in place. They're called the ciliary muscles. They have a pretty important job too. Their job is to be able to change the shape of this eye lens. Well, why do we need that? Well, we need that to change the power of our eyes depending upon how far the objects are that we are looking at. This will make sense if we draw some rays of light. So let's dim all of these things and consider some rays of light. Alright, imagine we are looking at objects which are very far away. Then the rays of light from that object will be parallel to each other. And in order to see it clearly, those rays must get focused onto the retina. So the ray diagram would look somewhat like this. Now the important thing is the amount of bending that is required. Notice that these rays have to be bent by this much amount so as to get focused on the retina. And this bending is done by this lens system, converging system. Now imagine that same object were to come closer. Again, to see it clearly, the rays of light must get focused onto the retina. So these rays will not change, but the incoming rays will now be diverging. And as a result, we will see now the required bending is higher. Alright, so just concentrate on the bending when the object comes closer. Here it is. Can you see that? The required bending is more. More refraction is needed, meaning more optical power is needed. So when objects are closer, we require more optical power, more bending. And when the objects are farther away, we require less optical power, less bending. So notice our eyes should be able to change its power depending upon how far the objects are. And that is accomplished by these ciliary muscles. So when the object is far away, the required power is less. And so the required curvature of our lens is also less. In such case, the ciliary muscles will be relaxed like what's shown over here. But when the objects come closer, since the required power is more, the ciliary muscles will start pushing on this lens to increase its curvature, as you can see. Like this. I've exaggerated the figure over here. It'll push on the lens, increase the curvature, and as a result, it will increase the optical power. And that's how, depending upon the the distance of the object from our eyes, the curvature of the lens will keep changing. And that is done by these ciliary muscles. And this phenomena, where ciliary muscles change the power of the eyes by changing the shape of the lens, is called accommodation. So that's their job. Their job is to perform accommodation. And by the way, we're going to look at accommodation in great detail in a future video. All right, so don't worry too much about this accommodation as of now. So that's pretty much it. So let's quickly summarize what we learned. We saw that the cornea, the aqueous humor, and the lens together form a converging system whose sole job is to focus rays of light onto the retina. The retina contains light-sensitive cells whose job is to convert light into electricity. These electrical signals are carried out by optic nerves. Their job is to carry the signal all the way to the brain so that the brain can process the information. The jelly-like substance, the vitreous humor, maintains the shape of our eyeball. The iris regulates the size of the pupil, which in turn regulates the amount of light that enters our eyes. And finally, the ciliary muscles regulate the power, optical power of our eyes, depending upon the viewing distance. Plants.
That was a great introduction to the anatomy of the eye. Does anyone have any questions from this particular video? Then we can tackle those first. I also have a quick recap for what we saw here. So here you can see the entire structure of the eye. The outermost layer is known as the cornea. It is the covering layer of the entire eye. Followed by we have a very condensed structure. It contains three parts. The first part is the iris that actually gives the color to the eye and covers the opening to the interior of the eye, and the pupil, which is the eye hole through which the light can enter. The third is the lens. This lens is created by multiple cells which express a protein known as crystallin, which allows a glass-like lens to form inside the eye. And through this, light can enter into the interior part of the eye where it will be detected by the retina. Retina is the back of the eye, which is filled up of neurons and sensory detectors, which are the rods and the cone cells that are present right here, embedded in this retina layer. And in between the lens and the retina, we have the vitreous humor, which fills this particular cavity. And between the cornea and the pupil, we have the layer, the aqueous humor, which fills this particular cavity. And the last thing that we need to be aware of is the ciliary muscles. So these muscles connect the lens to the rest of the eye, and these can contract or relax to actually either compress or elongate the lens. So depending on where how exactly the ciliary muscles are acting up, the lens would be either broader or lens would be either narrower and bigger in size. And depending if the lens is narrow or broad, then we have the power of this lens increases. So we have all heard of the term that our eyes, whenever someone is wearing glasses like me, they have power in their eyes. So what that means is that the normal power of this lens is hampered because of the impairment of the ciliary muscles right here. They either cannot relax enough or they cannot contract enough. If they cannot contract enough, then what happens is that the lens cannot become big enough or powerful enough or have enough power to focus close by objects. And if it cannot relax, then it cannot become thin enough to look at the objects at a distance. And that is where the two impairments of the eye, myopia and hyperopia, come into place. So this was a brief introduction into the anatomy of the eye. So these are all the structures that we looked at: cornea, pupil, iris, retina, the ciliary muscles, as well as the vitreous humor and the aqueous humor, which will mostly be important for all our discussions. So anyone has any questions from this? Okay. So as there are no questions, we go on to answer this question, which is the following part, not part of the eye. Retina is the neuronal component placed at the back of the eye. Cornea is the covering of the eye, which separates the pupil from the outside. Iris is the covering of the eye that prevents more or less light into the interior of the eye depending on the light conditions around us. And glomerulus, if you would know, is part of the kidneys. So the correct answer is kidneys. Okay, so we have two questions here. Aruna asks, what is the difference between myopia and hypermyopia? So I hope you're not confusing between myopia and hypermetropia. So if you're not, then myopia and hypermyopia would be myopic eyesight is when we are unable to relax our. So myopia is, so we have a slide on this. I'll quickly go to it. Uh, so you can see this, right? So myopia is when light is actually focused. So in order for us to see clearly, light must be converged or focused to the point on the retina itself. And that is a function of the ciliary muscles and the lens. But in many cases, what happens is that the lens or the ciliary muscles cannot focus the light onto the retina itself. If the light is converging somewhere ahead of the retina, we then then we can see blurriness in our eyes, and this causes myopia, where we cannot see things at a distance. If we have even higher levels of myopia, when the lens can relax even further, then the light rays will focus on at a distance even more ahead. So I think this is what you're referring to as hypermyopia. Is that okay, Aruna? And if you meant hypermetropia, then that is when the lens cannot focus on the retina, but the light rays converge somewhere behind the retina. That is the case which we know as farsightedness or hypermetropia, in which things that are right in front of us, those look blurry, but things further away from us look clear. That is because the the ciliary muscles cannot contract and force the lens force the lens to thicken and increase the power of the lens, so that the light rays will converge right on the retina. So the actual convergence occurs behind the retina, and we cannot see things very close to us. So that is known as hyperopia. And if we cannot see, we see bloody at a distance, but okay at nearby positions, then that is known as myopia, when the light rays converge ahead of the retina. Okay. So we had another question which Jagriti asks, hair cells are a part of eye or not? So hair cells are not part of the eye, those we do not consider as part of the eye. Eyes just consist of the parts that we have already seen. This is essentially the inner workings of the eye is what we consider as part of the eye. So everything inside the cornea is what we call the eye. Everything outside of it is part of the facial features. Is that okay, Jagruti? Okay. Moving on to the next question. The area from which optic nerve leaves The area from which the optic nerve leaves the retina is called the sensory spot, the blind spot, the retinal neuron area, or the descending region. Shivajit says it is B, the blind spot. Jagruti also says it is B, blind spot. Shantipriya also says it is B, blind spot. Divya and Vishwajit also says it is B, blind spot. Okay, let us look at it. So, if we look closely into the retina itself, as I said, there are many rod and cone cells embedded in the retina, and these are the light sensing elements of the eye. These rod and cone cells actually are arranged in a layer-like fashion. So you have rod and cone cells first, then we have another kind of cell which is known as a bipolar cell, then another kind of cell which is known as a horizontal cell, and finally you have another kind of cell which is known as a retinal ganglionic cell. So the rods and cones detect the light and then transfer the information one by one all the way to the retinal ganglionic cells. The retinal ganglionic cells are neurons which have very long axons, and the axons travel all the way from the surface of the retina to a hole in the back of the eye, where all the axons join together to form the optic nerve. The optic nerve is which carries the visual information to the brain. But if there is a hole in the back of the eye where the optic nerve is passing through, obviously there couldn't be any rods and cones in that area because it is just an empty space. So there are no rods and cones present right here. So if light falls on this area, then we will not be able to see this. And this is known as the blind spot. So the area from which the optic nerve leaves the retina is called the blind spot. Is that fine? I guess it's fine. The next question is, what part of the eye is majorly donated? The options are retina, cornea, iris, or glomerulus. Jakriti says it is B, cornea, which is mostly donated. Vishwajit also says it is B, cornea. Shantipriya also says it is B, cornea. Anyone else? Divya also says it is B, cornea. Okay, let us look at it. So all of you are again correct. The cornea, as we have said again and again, is the outermost part of the eye, and it covers the entire eye as well as the pupil. So it is obvious that the cornea should always be transparent in nature, so that it can allow light rays to pass through. But in case of certain diseases or trauma, this particular corneal layer can get cloudy or blurry, and which causes the entire eye to, like, you have low levels of blindness because of this. This can be fixed only. So there are two ways to fix this. The most relevant way that we have now is known as a corneal transplant surgery, in which we take a donor cornea and we actually remove this cloudy cornea from the patient and graft onto it the donor cornea, and then suture it onto the patient, and then after a while it functions as good as the original cornea. This kind of transplant surgery is very easy in the sense that most of the transplanted organs in our body have to continuously battle with the immune system in our body, because any foreign organ transplanted, such as say the heart or the kidneys, are always at the risk of immune rejection, because our immune cells recognize them as being foreign to our body and start attacking them. But in case of corneal transplant, And the good thing is that the cornea has very less blood transfer in it. So very less amount of immune cells actually are exposed to this foreign cornea, and you can go on ahead with your life with minimal risk of immune rejection, with the drugs being used to suppress whatever immune reaction there is. So, in the cornea is the most commonly grafted or transplanted part of the eye. What part of the eye is majorly donated? The answer is cornea. Is that okay? I guess so. Moving forward. Cataract is caused by the cloudiness of which of the following? The aqueous humor, the ciliary muscles, lens, or the vitreous humor? Jagruti says it is D, the vitreous humor gets cloudy in case of cataract. Anyone else? Shantipriya says it is C, the lens gets cloudy in case of cataract. Anyone else wants to try and answer this? Cataract is caused by cloudiness of which of the following? Aqueous humor, ciliary muscles, lens, or vitreous humor? Jagruti changes her answer to lens. Okay. So let us look at it. So cataract is the clouding of the lens. And obviously, as I have said previously, anything in the direction of the light path must be absolutely transparent to allow light to pass through. This is especially important in case of the lens, which is whose entire function is focusing the light onto particular regions of the retina. So when the lens gets cloudy, then we have possibility that the light will be blocked and not allowed to enter or focus properly on the back of the retina. And then we have cataract, in which there occurs blindness if left untreated. But the question is how exactly cataract is formed. To answer this, I would have to tell you how exactly the lens is formed. The lens is made out of multiple hundreds of cells which are arranged into this disc-like fashion, which essentially forms the structure of the lens. Initially, all of these cells, unlike normal cells, have their nuclei, mitochondria, Golgi body, everything else, and are functioning normally. But just before they are turned transparent, which occurs late in development, what happens is that they start expressing the lens protein crystalline. Crystalline is a protein which is allows the cell to completely turn transparent and allow light to pass through, which is followed by another protein uh, which is known as lipoxygenase. Lipoxygenase essentially dissolves almost all of the organelles of the cell, leaving behind just the crystalline protein and the cell membrane. Once all of the organelles are dissolved, the cell is essentially in a now a quiescent stage, not able to do any of its normal function, but is now essentially a transparent, completely transparent because of the accumulation of the huge quantities of crystalline protein. And all of these cells together are fixed in their position to form the lens. So this is an excellent organelle that is formed in our body through essentially programmed cell death. But now what exactly is cataract? Cataract occurs when the structure of the crystalline protein, so the structure of the crystalline protein is very important to allow the proteins to refract light in the way it does, for allowing it to become almost transparent. But in case of cataract, what happens is that due to old age or trauma again, the protein gets starting to misfold. They form different structures than their intended structures, which makes it so that they are not able to refract light like this and become opaque to the path of light. Once that happens, due to some unknown reason, this thing starts spreading from one lens to the other lens, one lens cell to the other lens cell. So that one by one, all of the crystallines present in all the cells start losing their function and became opaque. And this opacity is what is seen as the cloudiness in case of cataract. And once all the entire thing becomes cloudy, then we have complete blockage of light, which actually can cause near blindness in case of old people. The one of the ways to get around this is that you have heard of cataract surgery, in which a laser is used to disrupt or destroy the cells which have become opaque. Once those are removed, then the rest of the cells rearrange themselves to actually allow for light to pass through. And then you again have a clear path for light to pass through and eyesight can return. So this is how cataract works. So the options were cataract is caused by the cloudiness of the lens. Any questions from this? Okay, if there are no questions, we move forward. So a person with a 20/50 vision surely has which of the following? Myopia, hypermetropia, both A and B, or none of the above. Shantipriya says it is A myopia. Jagruti also says it is A myopia. Anyone else? Vishwajit also says it is A myopia. Okay. So Dibya also says it is A myopia. So you would probably have had, if you have had visits to the ophthalmologist, Banit also says A myopia by the way. So if you have had visits to an ophthalmologist or have had your specs added to your eyes, then you would surely know there is a term called a 20/20 vision, which is given to people who have almost perfect eyesight, which is equivalent to a normal eyesight. But can anyone tell me what exactly those numbers mean? 20/20, or in this case 20/50? Does anyone have any idea what exactly, how exactly does this code function? Like, what do you mean by a 20/20 vision? Can you take a guess? Anyone? No one has an idea what exactly the 220 stand for in a normal vision. So when we say 20/20 vision, what exactly it is? No one has any idea, right? Okay. So let me tell you, the 20s stand, the first 20 stands for something that is 20 feet away. And the next number indicates that how far away can you exact, like how far away was it kept apparently for you to look at it. So a 20/20 vision is that it was kept 20 feet away and you were able to see it 20 feet away. A 20/50 vision would mean that it was kept 20 feet away, but it appeared to be as blurry to you as if it was 50 feet away. So one of them is the real measure of the distance that the object is from you, and the next is the apparent measure of blurriness that you see because of the power in your eyes. So you probably would have seen a chart like this in the doctor's office. So all of these numbers, so if you start from here, a 20/20 would mean that this is small enough that it it is apparently placed at 20 feet away from you, and you can see it at that distance. So it is you have a 20/20 vision. A 20/25 vision would mean that it is 20 feet away actually, but you are able to see it as if it was 5 feet away. All of this is because the light rays from the objects are not able to converge at the right place on your retina. And that is why it appears to you as if it is further away, whereas it is actually closer than that. Similarly, 20/30, 20/40, 20/50 visions, actually you will be able to see this because you feel as if like your eyes detect it as being 50 feet away, whereas they're only 20 feet away. So that is a pretty bad condition. And this goes on, and this can go on in the opposite direction also. If an object is placed 20 feet away and you are able to read it very well, then your eyesight might be better than normal. So a 2016 score suggests that you're able to visualize the letters as if they were 16 feet away, but in reality they were 20 feet away. That just shows that your vision is better than 20/20 or the normal vision. This is the system normally used everywhere. You probably would have heard of a 6/6 vision as well. A 6/6 vision is what is used in case of some clinics and doctors. Here we are talking about the distance unit changes. So this is 6 meter by 6 meters, and here it was 20 feet by 20 feet. Both of them are equivalent, just you write it differently. Any questions from this? Okay. So here we are talking about myopia. We'll go through it again. So when a light enters your eye, the entire plan is that the lens will converge the light onto the retina. So this should be the focal point of the lens, and we want it to be focused here only. But in case of a myopic eye, what happens is that light coming from far away is not able to focus on the retina. This is because the lens and the ciliary muscles are contracted altogether into a more rigid state. So they are not able to relax and allow light to go even further and converge it somewhere ahead of the retina. This causes a blurry vision. And in case of highly myopic eyes, the lens is even more constricted by the ciliary muscles, and the lens converges the light even ahead of the retina, causes you to have extremely uh, blurry vision. And the way to fix this is that because the entire system acts as a convergent lens, which is acting more strongly than it should, then you add a concave lens on top of it, which combining both of them, what it will do is that it will cause the light rays to converge further away, because concave is divergent, so it actually pushes the focal point further away and allows it to land somewhere on the retina, allowing you to have clear vision. Okay. So a person with a 20/50 vision. So the person looks at something 20 feet away, but it looks blurry as if it was placed 50 feet away. That is the classic definition of nearsightedness, in which the light from far away cannot focus correctly on the back of your retina, and that is known as myopia. Is that all okay? Any questions? Does anyone have any questions from this? Okay. So as no one has any questions, we move forward. The focal length of the lens increases when ciliary muscles, the focal length of the lens, so the lens will now converge light at a distance further away from the retina when the ciliary muscles contract or are strained, relaxed, or the focal length of the lens becomes fixed, or the focal length does not depend on the ciliary muscles, but it depends on the size of the retina. Okay, Shanti Priya says it is B. When the ciliary muscles relax, then the focal length increases. Vishwajit also says B. And Jagriti also says it is B, relax. Anyone else wants to try? Dibya also says it is B. That the focal length of the lens increases when the ciliary muscles relax. Okay, so this is what it looks like. This is when you're looking at the eye from the top down with the light coming here. So these are the ciliary muscles, and the ciliary muscles can either contract and push the lens to allowing it to bulge, making the lens, the power of the lens even higher and allowing stuff close to you to converge at the back of your eye, or it can relax and the lens is pulled thin, the power of the lens goes down and you can focus something at something that is further away from you. So in case of focal length that increases, focal length increase in of ciliary muscles, the ciliary muscles relax. Any questions from this? Okay. If there are no questions, move on. For refractive power of lens to increase by 15 diopters, the shape of the lens changes from option A, very convex to very concave, option B, very concave to very convex, option C, very convex to moderately convex, and finally option D, moderately convex to very convex. So if the refractive power of the lens increases by 15 diopter, then does the lens become more convex or less convex? Anyone wants to try and answer this? Shantipriya says it is D, moderately convex to very convex. Jagriti also says it is D, moderately convex to very convex. Okay, let us look at it. So you probably have seen these light ray path diagrams previously when you have been doing physics in class 11 and 12. So what exactly is power of a lens? So the focal length of the lens is where parallel lights from an object will go, and if you hit the lens with parallel lights, then the focal length is the point where the lens will converge all the light rays. So this is known as the focal length. Power of a particular lens is 1 divided by the focal length in the meters unit. So the focal length should be expressed in meters before you do this, and the unit of power is diopters. So a convex lens, which will always focus the light rays on the right side. So after the light passes through it, will obviously always have a positive power, because focal length in this case is positive. A concave lens which diverges light, and causes the image to form on the left side before the light passes through the lens always has a power of negative. And the more powerful a lens is, the closer to it can converge. So because both the units, so both power and the focal length are inversely proportional, so shorter the focal length, more powerful more powerful the lens. So when we say that we are increasing the power by 15 diopters, we are essentially causing the light rays to converge even closer to the lens, and this can only happen if the lens has either become thicker or more powerful. So, or more convex, say, because a convex lens is known by the ability to converge light rays. So for a refractive power of the lens to increase by 15 diopters, so it is becoming more powerful. So that can only happen if the lens is becoming even more convex. So normally the power of a normal lens in our eyes is around 50 diopters. So it was already moderately convex and you are adding even more power on top of that. So it becomes very convex. So the correct answer is D, moderately convex to very convex. Any questions from this? Anyone wants to ask anything? Okay. Moving on, now we change gears and we'll be looking at the how exactly light is detected by the rod and cone cells. So the first question is, retina consists of photoreceptors, the rods and cones. Rods detect blank and cones can detect blank. So what can the rods detect and what can the cones detect? So the options are: rod detects the brightness of light, cones detect the intensity of light, rods detect the hue and the cones detect the color, rods detect hue, cones detect brightness, rods detect the intensity of light and cones detect the color of light. So we have a number of answers coming in. Shanti Priya and Jagruti both say D, it is intensity of the light and color of the light. Vishwajit and Surajit both also say it is D, that rods detect the intensity of light and cones detect the color of the light. Anyone else wants to try and answer this? Anyone else wants to try and answer this? Okay. So here again I have a video for you to look at. I'll just quickly play it. Let me know if it is audible.
Let's examine the difference between rods and cones in our eyes.
It's audible, right? Okay.
Let me draw a very simplified schematic of a rod just to give you an idea of what it looks like. So rods actually get their name because if you look at a rod under a microscope, it actually has this elongated cell body that kind of gives it a rod shape. So a rod is a photoreceptor. What exactly is a photoreceptor? Is it a neuron? Is it a type of nerve? So in fact, it is. It's a very specialized type of nerve that's able to take in light and convert it into a neural impulse. So inside a rod, there are a whole bunch of structures known as optic discs. And these optic discs are large membrane-bound structures inside the rods, and there are thousands of them in an individual rod. Embedded within the membrane of each optic
Yeah, I seem to have been. Hello, am I audible now?
Yes, sir.
Yeah, I got disconnected. Yeah, I think some of the people have left. I will just restart this. Yeah. Tell me if you can hear the video again.
Let's examine the difference between rods and cones
That is audible?
Yes, sir.
Okay.
in our eyes. Let me draw a very simplified schematic of a rod just to give you an idea of what it looks like. So rods actually get their name because if you look at a rod under a microscope, it actually has this elongated cell body that kind of gives it a rod shape. So a rod is a photoreceptor. What exactly is a photoreceptor? Is it a neuron? Is it a type of nerve? So in fact, it is. It's a very specialized type of nerve that's able to take in light and convert it into a neural impulse. So inside a rod, there are a whole bunch of structures known as optic discs. And these optic discs are large membrane-bound structures inside the rods, and there are thousands of them in an individual rod. Embedded within the membrane of each optic
Hello, can you hear me?
Yes, sir.
Okay, I think the video is not playing for some reason, so I will just explain it to you on the slides. Whoever is that, can you please mute yourself? Okay, so these are the rod cells that we are looking at. So rods have a particular structure in them, which is like a disc kind of structure, which go horizontally like this. These discs are regions where the protein, which is known as rhodopsin, is present. So you probably would have heard them the sense light using the protein rhodopsin. So rhodopsin is a very important protein. What happens is that when light falls on rhodopsin, so light is falling on rhodopsin, rhodopsin breaks into its two components. The two components individually are retinal and a second component which is known as opsin. So opsin is another protein which can go ahead and interact with another protein present on those disc-like structures, which is known as transducin. Why is all of this important? Because in our rods, there is continuously a increased polarization present in the rod. So rod cells are always at around minus 40 millivolts. And as we have seen earlier that around minus 40 volts if a neuron reaches, that it will give us a action potential. So the rods are always in a state where they continuously give us spikes of action potential. So completely you will see that if you electrophysiologically look at a neuron, it is always giving you spikes when in dark. But once in light, this entire pathway causes transducing to break cyclic GMP into normal GMP. GMP is guanidine monophosphate. So it goes from its cyclic form to its normal form. This conversion actually now hyperpolarizes the cell. So it goes from minus 40 to minus 80 millivolts. And once it hyperpolarizes, we know that it will not give us an action potential anymore. So essentially, in the presence of light, the neurons stop reacting or stop having an action potential. Every other time, they continue to have an action potential. So this could be seen by this image. So in the presence of light, in the absence of light, there is continuous cycling of sodium ions into the cell, into the neuron, because it is already at -40. So all the voltage sensitive, all the voltage sensing cationic channels are open, and current will keep on entering the cell, causing multiple action potentials. But in dark, but in the case of light, all these channels are closed off, and the neuron or the rod cell will not show any action potential. So initially when this was discovered, this was known as the dark current, because only in dark do we see the rod cell have any activity at all. So when there is darkness, the rod cell is firing action potentials. When there is light, the rod cell is not firing action potentials. So how exactly does this translate into actually looking or processing an image? So you have to understand, when light falls, so a rod cell turns off. But all the rod cells which is around it, where light is not falling, is still on. So it is very much like looking at a dark background, which is always dark, and a particular cell comes up and glows up as a light. So if you see something on a dark background, you see a light. So on a background of dark, you see a colored image of light because of this individual neurons switching off. Is that clear or no? Hello? Hello?
Sir, can you explain it once? I mean, once
One more
again.
time. Yes. So imagine all imagine you are looking at a whiteboard. Everything is white in color. So in that case, that is a situation corresponding to a retina where no light is falling. So all the neurons are firing. So everything is white. But the moment light falls on a neuron, that particular rod cell now stops showing action potential. So you will have a black spot in that particular region because that neuron is not firing anymore. So if you have an image, say, of a person standing in front of you, so like a person standing in front of you, all the rod cells which are not looking at the light coming from that person will continue firing. But once the rod cells which will look at the light coming from the person will turn off. So you will see a dark outline of that person on a bright background. That is how the entire retinal circuit works. So you have a background which is always firing, and only where the light falls, that particular part turns off. So you can see things on a bright background but having a black image. Is that okay? Uh,
No, sir.
okay, okay. Let me try something. Can you see my screen?
Yes, sir.
Okay. So imagine that this entire thing is the retina, where white corresponds to a particular rod cell firing. So in the dark, when there is, so let me show it in a different way. Okay. So if you have a dark background such as this, where you can first imagine that in the dark background, all the rod cells were firing. And then you have a particular region where none of the rod cells are firing, such as this. Say you are looking at the light coming from just this 7 figure. When light coming from this particular figure hits your eye, these neurons or these rod cells which were looking at this particular light will stop firing, but everything else is still firing. So on a dark background, you will see a white image. So every one of the other rod cells is still firing and gives you a black background. But this particular light falling on these rod cells will stop them from firing, and that particular image will emerge out of them. Because everything else is firing, they are not, so you can see the image. Is that okay? So if you see the opposite situation, the opposite situation is that you have a white background where you are looking at a dark image. So every one of the rod cells around these particular cells are firing, and every one of the cells where this particular light is falling are turning themselves off. So you can see all of these off cells as the black line and everything else in the background as white.
Okay, sir.
Okay. Okay, so retina consists of photoreceptors, rods, and cones. Rods detect the intensity of the light, and cones detect the color of the light. So, rods, as we have seen, can react to any light of any wavelength. Any light of any wavelength falls on them, and then they stop firing. But cones, they are sensitive to the wavelength of light. So, cones can either be red cones, blue cones, or green cones, the RGB spectrum. So, red cone will only fire in case of red light or stop firing in case of a red light. Green cone will stop firing in case of a blue light. A green light and blue cone will stop firing when blue light is shined on it. So, using this all these colors, we can make up almost all the colors that they are in the world, and we only and thus cones are able to detect the color of light because they are only sensitive to that particular wavelength of light falling on them, whereas rods are sensitive to any light and detect the intensity of light. Okay, the next is in the dark the photoreceptor is depolarized due to an influx of blank through open ion channels that are gated with blank. So in the dark, the photoreceptors are always depolarized due to influx of which particular ion, and the entry of this ion is stopped by which of the channels. So the options are sodium and calcium, CGMP, sodium potassium calcium, potassium CGMP calcium, potassium and sodium. So Vishwajit and Jagruti both say it is A, sodium and calcium. We have influx of sodium and calcium, and the channels are gated using cyclic GMP. Okay, so let's see if this video plays or not. Let me know if this is audible.
Last video, we looked at how light from the sun can enter the eye, and this is the eye, so it enters the eye, hits the back of the eye, and somehow through a set of steps it gets converted into neural impulse, and the impulse gets sent to the brain for you to make sense of the information. So we looked at something called the retina in the last video, and we talked about how the retina is made up of a bunch of different cells, and the two main cells are rods and cones. So we're going to look at just the rods in this video. So the rod, which is shaped like this, let's give him a little smiley face because he's happy. And what he does is that as soon as light is presented to him, he basically takes that light and then converts it into a neural impulse. So normally he is turned on. When there's no light, the rod is turned on. But when light is present, it actually turns him off. So with light, he's turned off. So how does this happen? So this occurs through this thing called the phototransduction cascade. So the phototransduction cascade is this set of steps that occurs at the molecular level that basically takes this rod and turns him off. And in turning him off, he's actually able to turn on a bunch of other cells that eventually lets the brain know, hey, there's light here. So it allows the brain to realize that there's light and allows it to comprehend what's going on and make sense of the world. So let's go ahead and examine this phototransduction cascade. So let's give ourselves a little bit of space here to just look at it in a little bit more detail. So let's look at just a part of the rod. So I'm going to redraw the rod over here, and let's just look at this part of the rod, just this very top little bit of the rod. And I'm going to draw it a lot bigger. I'm going to draw it a lot bigger so that we can really make sense of what's going on here. So inside the rod, which we just made much bigger, there are a bunch of these little discs. So they're really thin little discs, and they're just stacked on top of one another, and there are hundreds of them. And they're like this. They just stacked on top of one another, and they fill up the entire rod. Same thing in cones, but we're just going to look at the rods here. So inside of these little discs, there are a whole bunch of different proteins, all interspersed throughout the discs. So this protein that I'm drawing in red, let me just go ahead and draw it a lot bigger. So we're going to go ahead and blow this protein up. And it's basically this multimeric protein. So it consists of a bunch of different subunits. There are actually 7 subunits. So there are 5, 6, and 7. So there are all these subunits that make up this little protein right here. So this protein as a whole is called rhodopsin. And it's called rhodopsin because it's in a rod. If it were in a cone, it would be called conopsin, but it's basically the same protein. And sitting inside of this protein is a small molecule, and it sits just inside. Here, let's go ahead and just kind of just zoom in a little bit just so that we can make a little bit more sense of what we're looking at here. So let's go ahead and zoom in over here. So this little molecule is just sitting inside of rhodopsin, and it's kind of bent. You can see here how I drew it. It's just a little bit bent. So this little molecule is called retinal. Retinal. And in this conformation, in this bent conformation, we call it 11-cis retinal. And so what happens is light comes in from the sun, goes in through the pupil, hits the retina, and then hits this little rod. Some of the light will actually hit this molecule. So it'll hit rhodopsin, and then it'll actually hit this molecule. So here's the light wave. It comes in, and it just hits the molecule right there. So an interesting thing happens when the light hits the molecule right at this region. And what it actually causes is it causes the retinal to actually change conformation. So it actually causes the light provides enough energy that the retinal goes from this bent conformation, and it causes it to look more like this. So I'll draw it over here. Draw a little carbon ring so it becomes straight. So it went from being bent to being straight. So the light does this, and basically it goes from being 11-cis retinal to all trans retinal. So when the retinal changes shape, it actually causes the rhodopsin molecule to also change shape. So the two are really closely linked. So when the retinal changes shape, the rhodopsin changes shape. So let's go ahead and pretend that it looks like this. So this region that I'm shading in right here is what the new rhodopsin looks like after the retinal has changed shape after the light hit it. So rhodopsin now changes shape, and that basically begins this big cascade of events. So what happens next is there's a molecule, and I'm going to draw that molecule right here in green, and it's made up of 3 different parts. So there's this alpha subunit, there's a beta subunit, and there's a gamma subunit. And this molecule as a whole is called transducin. So transducin. Oops. All right. Sorry here. So transducin. So transducin basically, as soon as retinal changes shape and causes rhodopsin to change shape, transducin breaks away from rhodopsin, and the alpha subunit actually comes over here to another part of the disc and binds to a protein called phosphodiesterase, which I'll just draw as this little box over here. So this little protein is called cyclic GMP phosphodiesterase, or PDE for short. So PDE, basically what it does, let me just go ahead and zoom out a little bit. What PDE does when it's activated is it takes cyclic GMP, which is floating all around the cell. It's a little molecule, it's tiny, and it basically takes the cyclic GMP and converts it into just regular GMP. So this basically reduces the concentration of cyclic GMP and increases the concentration of GMP. And the reason that this is important is because there's another channel over here. So there's a whole bunch of these sodium channels. So a whole bunch of sodium channels, and they're all over the cell. So they're just a whole bunch of them. And basically what they let the cell to do is they allow the cell to take in sodium from the outside. So let's just say there's a little sodium ion, and it allows it to come inside the cell. So in order for this sodium channel to be open, it actually needs cyclic GMP to be bound to it. So as long as cyclic GMP is bound, the channel is open. But as the concentration of cyclic GMP decreases because of the phosphodiesterase, it actually causes sodium channels to close. So now we have a closing of sodium channels, and now we basically have less sodium entering the cell. And as less sodium enters the cell, it actually causes the cell to hyperpolarize and turn off. So as the sodium channels close, it actually causes the rods to turn off. So turn off. So basically without light, the rods are on because these sodium channels are open. Sodium's flowing through and the rods are turned on. They can actually produce an action potential and activate the next cell and so on. But as soon as they're turned off, what happens is very interesting, because let's just look at this rod over here. So what happens is really interesting because there's this other cell over here that is called the bipolar cell, and we'll just give it kind of a neutral face because it's bipolar. This cell, there are actually two different variants. So there are on-center and there are off-center bipolar cells. Off-center. So the on-center bipolar cells normally are being turned off when this rod cell is turned on. But as we mentioned, due to the phototransduction cascade, the rods turn off, which actually turns on the bipolar cell. So basically, on-center bipolar cells get turned on with light and get turned off when there's no light. So that's how they get their names. So when the bipolar cell gets turned on, it activates a retinal ganglion cell, which then sends an axon to the optic nerve and then into the brain. And so that process is known as the phototransduction cascade, and it basically allows your brain to recognize that there's light entering the eyeball.
Okay, does the entire thing make more sense now? After looking at how rods and cones function. Hello? Okay, so maybe I would encourage you to go and look at the uh, videos from this particular week, and Professor Mehna has actually gone through this particular thing in a bit more detail, so maybe that will actually help you to understand. So coming back to this, in the dark the photoreceptor is depolarized due to influx of sodium and calcium open through ion channels that are gated by cyclic GMPs, what we just saw. Okay. Night blindness is caused by loss of vitamin A, which leads to a decrease in which of the following? Retinal, vitamin D, melanin, or cone cells. Jagriti says it is A, retinal. Anyone else? Shantipi also says it is A, retinal. Okay, let us look at it. So as I said, in the entire bleaching cycle of the visual cycle, one of the most important molecules is retinal, because it actually helps in detection of the light. Without the light actually hitting the rhodopsin molecule and being transferred into retinal and opsin, we will not be able to actually detect the light rays. And most of the time night blindness is because there is a decrease in the amount of retinal. But where does retinal come from? Retinal is made from retinol. Where does retinol come from? Retinol comes from the vitamin A that we consume.
Sir,
Yes.
sir, screen is not I mean, the
Okay, okay, okay.
it is not visible.
Okay, okay, okay. Screen is not visible. Is it visible now? Is it okay? Can you see the screen now?
Yes, sir.
Okay, so this was the question. Night blindness is caused by loss of vitamin A, which leads to decrease in which of the following? Retinal, vitamin D, melanin, or cones. So as I was talking, that the all important molecule for sensing light is rhodopsin. So one of the most important parts of rhodopsin is this molecule known as retinal, which actually dissociates the rhodopsin into retinal and opsin and starts the downstream cascade. So, but we have to know where the retinal comes from. Retinal comes from retinol, and retinol is actually converted from vitamin A that we consume. So that is the source of retinol, which in turn gets converted to retinal and incorporated into rhodopsin. So no vitamin A, you will have very poor eyesight. So you would probably have heard that our parents and older people always ask us to eat carrots to improve our eyesight. The reason is that carrots are full of keratin, uh, not keratin, carotene. So similar pronunciation, different spelling, which is a precursor to vitamin A, and which in turn produces retinol once it enters our body and allows more retinal to be produced. So more retinal, more rhodopsin, better sensing of light, and that is what enables us to look and detect light even in low light conditions in case of night. And thus night blindness is caused when there is a loss of vitamin A in our diet, which is due to a decrease in retinal. Any questions from this?
No, sir.
Okay, so that's all I had for you today. If no one has any questions, then we can end the session for today. Okay. Thank you everyone for joining, and I will see you next week. Thank you.